മാസ്റ്റർ കീ അക്കാഡമിയുടെ പുതിയൊരു ഓൺലൈൻ സെക്ഷനിലേക്ക് നിങ്ങൾക്ക് ഏവർക്കും സ്വാഗതം. ഇന്നിപ്പോൾ നമ്മൾ പഠിക്കാൻ പോകുന്ന ടോപ്പിക്ക് സ്റ്റെയിനിങ് ടെക്നിക്സ് ഇൻ മൈക്രോബയോളജി എന്നതാണ്. അപ്പോൾ സ്റ്റെയിനിങ് ടെക്നിക്സ് ഇൻ മൈക്രോബയോളജിയിലേക്ക് നമ്മൾ കടക്കുന്നതിന് മുമ്പായിട്ട് നമുക്ക് ബാക്ടീരിയൽ സെൽ സ്ട്രക്ചർ എന്താണെന്നൊന്ന് നോക്കാം നമുക്ക്. So the bacterial cell structure, it is organized into three categories, that is internal structure, cell envelope and external structure. Internal cell structures include cytoplasm, nucleoid, bacterial chromosome, plasmid, ribosomes and storage granules. Cell envelope includes cell membrane and cell wall. Cell wall includes peptidoglycan cell wall and outer lipid membrane cell wall. That is only in gram-negative bacteria. And external cell structures which include appendages and covering, that includes flagella, fimbriae, sex pilus and glycocalyx. The staining techniques, അത് മെയിനായിട്ടും നമ്മൾ ഡിസ്ക്രൈബ് ചെയ്യുന്നത് that is on the basis of the cell wall. അപ്പം നമ്മൾ ഇവിടെ ഇമ്പോർട്ടൻസ് കൊടുത്ത് നമ്മൾ എക്സ്പ്ലെയിൻ ചെയ്യാൻ പോകുന്നത് ബാക്ടീരിയൽ സെൽ വാളിനെ പറ്റിയാണ്. സോ ദ സെൽ വാൾ ഇറ്റ് ഈസ് ഡിവൈഡഡ് ഇൻ ടു ടു ടൈപ്പ്, ദാറ്റ് ഈസ് ഗ്രാം പോസിറ്റീവ് സെൽ വാൾ ആൻഡ് ഗ്രാം നെഗറ്റീവ് സെൽ വാൾ. ഗ്രാം പോസിറ്റീവ് സെൽ വാൾ, സോ ബാക്ടീരിയൽ സെൽ വാൾ എന്ന് പറഞ്ഞാൽ ഇറ്റ് ഈസ് ദ ലെയർ ഓഫ് ദ സെൽ എൻവലപ്പ് that is lying between the cytoplasmic membrane and the cell envelope. It is known as the cell wall, and it is in gram-positive bacteria. It is made up of Peptidoglycan and Teichoic acids. Gram-positively, it is made up of Teichoic acid and Peptidoglycan. Then about the functions of the cell wall, the bacterial cell wall. The main function is it provides structural integrity to the cell. The bacterial cell wall, it differs from the another organisms by the presence of peptidoglycan, which is located immediately outside the cell membrane or the cytoplasmic membrane. Peptidoglycan is responsible for the rigidity of the bacterial cell wall and for the determination of cell shape. So mainly the cell wall, it is important for maintaining the cell shape and integrity. And the cell wall, it is relatively porous and is not considered to be a permeability barrier for small substrate. While all bacterial cell wall, with few exception, for example intracellular parasite such as Mycoplasma, that's very important. Mycoplasma is cell wall deficient bacteria that contain peptidoglycan. Not all cell wall have the same overall structures. This is notably expressed through the classification into gram-positive and gram-negative bacteria. So the main thing that differentiates gram-positive and gram-negative is the composition of the cell wall. So this is the structure of bacteria, and this is the cytoplasm. This is the cytoplasm, and this is the nucleoid. Then about the outermost layer, that is the capsule. Then this is the cell wall. This one. This is the cell wall. Then cytoplasmic membrane, and internally there is the ribosomes, and there are certain thread-like appendages that is known as pili or fimbriae, and another thread-like appendage that is the flagella, which is the locomotory organ of bacteria. And that is the anatomy of a bacterial cell. So first about gram-positive cell wall. Gram-positive cell wall, it is usually thick compared to gram-negative. It is usually thick and it is homogeneous. And the cell wall is mainly, in gram-positive, it is mainly made up of peptidoglycan. And the peptidoglycan of gram-positive cell wall that constitute about 50 to 90% of the total dry weight of the cell wall. And in addition to peptidoglycan, it contains large amount of teichoic acid. These teichoic acids are the polymers of glycerol or ribitol joined by phosphate group. So gram-positive cell wall is mainly made up of peptidoglycan, and in addition it contains teichoic acids. Then we have to see what is peptidoglycan. Peptidoglycan, it is also known as murine. The another name for peptidoglycan is murine. That is a polymer consisting of sugars and amino acids that form, the peptidoglycan forms a mesh-like layer outside the plasma membrane of bacteria forming the cell wall. And the peptidoglycan, it consists of three components or three parts. That is a backbone. The backbone of the peptidoglycan is two amino sugars, that is N-acetyl glucosamine and N-acetyl muramic acid, which forms the backbone of peptidoglycan. And in addition to N-acetyl glucosamine and N-acetyl muramic acid, it is commonly known as NAG and Na. And in addition, it contains alternating chains of amino acids, alternating chain of amino acids, that is that chain may be, that amino acid chain is known as peptide chain, that is tri, tetra or pentapeptide chain. So this is the structure of peptidoglycan. This is the structure. It consists of two amino sugars, that is N-acetyl muramic acid and N-acetyl glucosamine. This N-acetyl muramic acid and N-acetyl glucosamine, they are arranged alternatively. And there are three parts. And already പറഞ്ഞു, മൂന്ന് parts ആണുള്ളത്. N-acetyl muramic acid and N-acetyl glucosamine, and in addition it has a peptide chain constituting amino acids. The peptide chain, it is connected to N-acetyl muramic acid. This is connected to, this is the chemical structure, and this is the NAM and NAC. This tetrapeptide chain, this is the tetrapeptide chain containing 4 amino acids, and this is connected to NAM molecule. And the 4 amino acids are L-alanine in position 1, D-glutamic acid in position 2, then mesodiaminopymalic acid in position 3, and D-alanine in position 4. And here the L and D amino acids, they are arranged alternatively. In the first position is always L-alanine, which is connected to NAM molecule, which is connected to the carboxyl group of the NAM molecule. And second D, third one again L or mesodiaminopymalic acid, and fourth one is D-alanine. And these tetrapeptides, the neighboring tetrapeptides, this is the NAM molecule of first chain, and this is the NAM molecule of the second chain, and the neighboring tetrapeptides, they are connected by a bond, and that bond is pentaglycine peptide interbridge. The bond is pentaglycine peptide interbridge. The neighboring tetrapeptides. And the neighboring tetrapeptides is connected by pentaglycine peptide bond, which is formed between the third amino acid. Which is the third amino acid? That is mesodiaminopymalic acid. Third amino acid of the first NAM molecule, and the fourth amino acid, that is L-alanine of the neighboring tetrapeptide. So the bond is formed between the third and the fourth amino acids. And the bond is pentaglycine peptide interbridge. Okay. So you can see here the bond. This is the pentaglycine peptide bond. So ഞാൻ പറഞ്ഞു നിങ്ങളോട് gram-positive cell wall is mainly made up of peptidoglycan, and in addition to peptidoglycan, gram-positive cell wall contains teichoic acids. The teichoic acids, they are connected to either peptidoglycan or to plasma membrane lipids. These teichoic acids are polymers containing ribitol or glycerol. And these teichoic acids, they may be connected to peptidoglycan or they may be connected to plasma membrane, the underlying plasma membrane. Beneath the cell wall, there is the plasma membrane, and the teichoic acids may be connected to peptidoglycan or to the underlying plasma membrane lipids. So, if it is connected to peptidoglycan, it is known as wall teichoic acid. And if it is connected to plasma membrane lipids, it is known as lipoteichoic acids. So the teichoic acids may be of two types, lipoteichoic acids and wall teichoic acids. And these teichoic acids, they are absent in gram-negative bacteria. Then about the functions of the teichoic acids. These teichoic acids are antigenic determinants. That is, they act as the receptor molecule for bacteriophage. Then they participate in the supply of magnesium to the cell by binding magnesium ions. They regulate normal cell division. Then for most part, protein is not found as a constituent of gram-positive cell wall except M protein on group Streptococci. So this protein normally gram-positive cell wall il കാണുന്നതല്ല. പക്ഷെങ്കിൽ Streptococci, that's a gram-positive bacteria. Streptococciയുടെ cell wall നമ്മൾ നോക്കിക്കഴിഞ്ഞാൽ that contains a type of protein and that is M protein. This is the structure of gram-positive cell wall. So this is the peptidoglycan layer that forms the main layer that constitute about 50 to 90%. And in addition to type of teichoic acids, that is lipoteichoic acids and wall teichoic acids. Lipoteichoic acids are those which are connected to underlying plasma membrane lipid. This is the beneath the peptidoglycan, beneath the cell wall or peptidoglycan layer, there is the plasma membrane. So these lipoteichoic acids are those which is connected to cytoplasmic membrane lipids, plasma membrane lipids. And whereas the wall teichoic acid, it is connected to, this is the wall teichoic acid. This wall teichoic acid is connected to peptidoglycan, and these are the wall associated proteins. And this is about the peptidoglycan synthesis. gram-positive peptidoglycan synthesis നടക്കുന്നത് എങ്ങനെയാണ് എന്നുള്ള ഡയഗ്രാം ആണ് ഈ കാണിച്ചിരിക്കുന്നത്. And in the synthesis of peptidoglycan, there are two type of carrier molecules are involved, that is the bactoprenol and another one is the UDP. So the synthesis involve two type of carriers, bactoprenol carrier and UDP carrier. This UDP, it is the carrier of the N-acetyl muramic acid and N-acetyl glucosamine which is synthesized in the cytoplasm. And bactoprenol, the function of bactoprenol is to transport the peptidoglycan components across the membrane. Okay. So this is the comparison of gram-positive cell wall and gram-negative cell wall. So the diagrammatic representation. This is the plasma membrane, and in between the plasma membrane and the cell wall there is a space that is known as periplasmic space. This periplasmic space is not so clear in gram-positive bacteria. This green color that indicates the cell membrane or plasma membrane. This light blue color, that is the periplasmic space, that is not so clear in gram-positive. And outer to the plasma membrane there is the cell wall, which is made up of peptidoglycan and teichoic acids. Whereas in gram-negative bacteria there is a clear periplasmic space. ഈ ഡയഗ്രത്തിൽ നോക്കിക്കഴിഞ്ഞാൽ നമുക്ക് അത് വ്യക്തമായിട്ട് മനസ്സിലാകും. This periplasmic space is not very clear, but here the periplasmic space in gram-negative bacteria, the periplasmic space is very clear. Then about gram-negative cell wall. Gram-negative cell wall compared to gram-positive bacteria, it is very complex than gram-positive. And this gram-negative cell wall, it is made up of two layers, an outer membrane and a peptidoglycan layer. The peptidoglycan layer, ഈ പടത്തിൽ നോക്കിക്കഴിഞ്ഞാൽ അത് ക്ലിയർ ആകും. പ്ലാസ്മ മെംബ്രെയിൻ, then the periplasmic space, and this is the outer membrane. In between the outer membrane and plasma membrane there is the periplasmic space, and within the periplasmic space lies the peptidoglycan, and the outer membrane lies outside the peptidoglycan layer. So the difference, gram positive peptidoglycan is the outermost layer, it forms the cell wall. But gram negative നമ്മൾ cell wall നോക്കിക്കഴിഞ്ഞാൽ the cell wall contains two components, an outer membrane and a peptidoglycan layer. gram negative രണ്ട് layers ആണ്, outer membrane ഉം peptidoglycan layer. അതിൽ peptidoglycan layer എന്ന് പറയുന്നത് അത് പെരിപ്ലാസ്മിക് സ്പേസിലാണ് കാണപ്പെടുന്നത്. എന്നാൽ ഗ്രാം പോസിറ്റീവിൽ പെരിപ്ലാസ്മിക് സ്പേസ് വളരെ ചെറിയൊരു പെരിപ്ലാസ്മിക് സ്പേസ് ആണുള്ളത്. അതിനും പുറത്തായിട്ട് ഒരു തിക്ക് ലെയറായിട്ട് പെപ്റ്റിഡോഗ്ലൈക്കൺ കാണപ്പെടുന്നു. അപ്പം gram negative cell wall, it contains an outer membrane and peptidoglycan. So the difference between the peptidoglycan of gram positive and gram negative, peptidoglycan lies in the periplasmic space and it constitutes only about 5 to 10% of the total dry weight of the cell wall. Whereas in gram positive bacteria, the peptidoglycan, it constitutes about 50 to 90% of the total dry weight of the cell. Whereas here it is only about 5 to 10% of the total dry weight. And another difference is there is in between the tetrapeptide chain, in between the amino acids. In gram positive bacteria, the tetrapeptide chain are connected by pentaglycine peptide bond, whereas in gram negative bacteria that pentaglycine bond is absent. Instead there is the direct linkage. There is a direct linkage between neighboring tetrapeptides. Okay, but difference, peptidoglycan, it's a very thin layer gram negative, whereas it is a very thick layer in gram positive bacteria. And the cell wall of gram negative has an outer membrane and a peptidoglycan layer. Then the outer membrane, it lies the thin outside the thin peptidoglycan. Outer membrane is present outer to the peptidoglycan, and the outer membrane is made up of lipopolysaccharide and proteins. This is the outer membrane. This outer membrane it lies outside the thin peptidoglycan layer. This is the outer membrane. And this outer membrane, it is made up of lipopolysaccharide and proteins. So outer membrane structure നോക്കാം നമുക്ക്. Outer membrane structure is made up of proteins and lipopolysaccharides. So first about the outer membrane proteins. So mainly there are 4 types of proteins, that is bronze lipoprotein, matrix protein, protein 2 and band 3 protein. Bronze lipoprotein, that is the most abundant lipoprotein or the most abundant protein in gram negative cell wall is bronze lipoprotein. This bronze lipoprotein, it is covalently linked to the underlying peptidoglycan. Outer membrane is present outer to the peptidoglycan. So this bronze lipoprotein, it is covalently linked to underlying peptidoglycan, and it is embedded in the outer membrane by its hydrophobic end. And because of this covalent linkage, this outer membrane and peptidoglycan is firmly linked so that it can be isolated as a single unit. This outer membrane and peptidoglycan, it is not present as a separate structure, it is present as a single unit, and this is by the linkage of this bronze lipoprotein. Then the second protein, that is the matrix protein, it is non-covalently associated with the peptidoglycan. And this matrix proteins, it form passive diffusion pores. So these matrix proteins are also known as porine proteins. Matrix proteins are also known as porine proteins. And in the structure, നമുക്ക് കാണാം 3 matrix proteins, they cluster together and they form a channel. The third protein is protein 2, and it is closely related to matrix protein. It is having the same structure as that of matrix protein. And the band 3 protein, it is exposed to outer surface of the membrane. So that is about the outer membrane proteins. The outer membrane is made up of proteins, and the second one is the lipopolysaccharide. The main component of the outer membrane is lipopolysaccharide. The lipopolysaccharide molecule is extremely important from a medical standpoint, and it consists of 3 parts, that is lipid a core polysaccharide and O antigens. The lipid A, that part is embedded in the outer membrane. The core polysaccharide, it is located on the surface of the membrane, and the O antigens, which are short polysaccharides extended out from core. Okay. So it is made up of 3 components: lipid A, core polysaccharide and O antigen side chain. First, lipid A. This lipid A, it is the carbohydrate component of the lipopolysaccharide. And this lipid A is made up of 2 glucosamine molecules. That is, it is made up of a beta 1,6-D glucosamine disaccharide. And it also, in addition to this glucosamine disaccharide, it contains a polysaccharide chain extending from one of the position, then a phosphate chain. That phosphate may be pyrophosphate also, and fatty acids. And there are 6 long chain fatty acids. They are lauric acid, palmitic acid and myristic acid in the ratio 1:1:1. That is, അതായത് 1 lauric acid, 1 molecule of palmitic acid and 1 molecule of myristic acid. And also it has 3 molecules of hydromyristic acid. So there are totally 6 long chain fatty acids. The lipid A, that contains glucosamine, polysaccharide, phosphate and 6 fatty acids. Then about the polysaccharide chain. The polysaccharide chain contains two regions, an inner core and an outer core. This inner core is made up of two regions, a KDO region and a dihaptose region. This KDO region that contains 3 molecules of an 8-carbon sugar, that is keto-deoxyoctanoate, and the dihaptose region that contains 2 molecules of a 7-carbon sugar, that is L-glycero-D-manoheptose. And the outer core, it has 2 regions, inner and outer. Then the outer core is also made up of 5 sugars, that is 2 glucose, 2 galactose and 1 molecule of N-acetyl glucose amine. So totally the inner and the outer core that contains 10 sugar molecules, 5 inner core and the next 5 outer core. Then the third part, that is the O antigen side chain, and that O antigen side chain, it is a very short polysaccharide chain and it is different in different species of bacteria. Okay. This is about the structure of, this is the diagram of gram-negative cell wall. Gram-negative cell wall, ഞാൻ പറഞ്ഞു, that contains a peptidoglycan layer and an outer membrane. This peptidoglycan layer, this is the periplasmic space, and the peptidoglycan layer is a very thin layer that lies in the periplasmic space, and outer to the peptidoglycan layer there is the outer membrane, which is made up of proteins and lipopolysaccharide. These are the proteins. This is the lipoprotein and this is the matrix protein. Matrix proteins are also known as porine proteins, and 3 porine proteins or matrix proteins, they cluster together and they form a channel, and this lipoprotein, they connect the peptidoglycan to the outer membrane. This is the connection, so that it is isolated as a single unit, and beneath the cell wall or the peptidoglycan layer there is the plasma membrane. So this is the diagram of gram-negative cell wall. This is the comparative chart of gram-positive and gram-negative. നമ്മളിപ്പം gram-negative cell wall gram-positive cell wall നമ്മൾ സ്ട്രക്ചർ നോക്കിക്കഴിഞ്ഞു. അപ്പോൾ അത് രണ്ടും കൂടെ നമുക്കൊന്ന് കമ്പയർ ചെയ്ത് നോക്കാം. ഫസ്റ്റ് about the thickness. Gram-positive, the thickness is 20 to 80 nanometer. In gram-negative, the thickness is 10 nanometer. Number of layers in the wall, only 1, and here it is 2. That is, നമുക്കറിയാം gram-positive cell wall is only made up of peptidoglycan. അവിടെ peptidoglycan layer മാത്രമേ ഉള്ളൂ. ഇവിടെ 2 layers ഉണ്ട്, outer membrane ഉം peptidoglycan layer. Peptidoglycan content very thick layer ആണ് gram-positive എന്ന് നമ്മൾ പഠിച്ചു. That is more than, greater than 50%. It is only 10 to 20%. Teichoic acids gram-positive മാത്രമേ present ആയിട്ടുള്ളൂ, gram-negative teichoic acids absent ആണ്. Lipid and lipoprotein content വളരെ percentage കുറവാണ് positive, only about 0 to 3%. എന്നാൽ gram-negative it is about 58%. പ്രോട്ടീൻ കണ്ടന്റ് നോക്കിക്കഴിഞ്ഞാലും അതുപോലെ തന്നെയാണ്, പോസിറ്റീവിൽ 0% ആണ്, ഇറ്റ് ഈസ് 9%. ലിപ്പോപോളിസാക്കറൈഡ് 0, in gram-negative 13%. And about the sensitivity to penicillin antibiotic, it is very sensitive, gram-positive is very sensitive to penicillin, whereas it is not as much in gram-negative. ആൻഡ് ഡൈജഷൻ ബൈ ലൈസോസൈം എൻസൈം. ദിസ് ഗ്രാം പോസിറ്റീവ് സെൽസ് ആർ ഈസിലി ഡൈജസ്റ്റഡ് ബൈ, പെട്ടെന്ന് നമുക്ക് ഡൈജസ്റ്റ് ചെയ്യാവുന്നതാണ് ലൈസോസൈം വെച്ചിട്ട് ഗ്രാം പോസിറ്റീവ്. ബട്ട് അത്രയും ഗ്രാം നെഗറ്റീവിൽ കാണത്തില്ല. ഓക്കെ.